Continuing on, looking at acids and bases. In aqueous solution, an acid dissociates into hydrogen ions and anions. Nearly every molecule of a strong acid dissociates, producing a high concentration of hydrogen ions. In aqueous solution, a base dissociates into hydroxyl ions and cations. Nearly every molecule of a strong base dissociates, producing a high concentration of hydroxyl ions. Now on the pH scale, 7 is going to be neutral. Any number lower than 7 is going to be an acid. Any number higher than 7 is going to be an alkaline or a base. And just some examples. Arterial blood, for instance, has a pH of about 7.4. And as we go farther down with a lower number, okay, we can see that, for instance, milk, urine, and saliva is about 6.3 to 6.6, with black coffee at about 5. And then grapefruit juice, soda, tomato juice, 2.5 to 3.5. Lemon juice and vinegar is about 2. And then finally at 0, we have battery acid and hydrochloric acid. The interesting thing is hydrochloric acid is made and found in the stomach. And then going the opposite way, if we're going higher than 7, we're becoming more basic or alkaline. Seawater has a pH of about 8. Baking soda, about 9.5. Ammonia solution, 10.5 to 11.5. Bleaches, oven cleaners, lye, very caustic, 13.5. And liquid drain cleaner with a pH of about 14. And so you can get an idea, at either extreme, they can be very dangerous and can burn you, okay? But again, just so you know, 7 is neutral. The number greater than 7 is alkali or basic. And lower than 7 is going to be toward the acid side. Now looking at monosaccharides, these are the 5 important monosaccharides: glucose, fructose, galactose, and deoxyribose and ribose. Now deoxyribose is going to be found in DNA, and ribose is going to be found in RNA. Now some of the important disaccharides, and again, di is 2, saccharide is sugar, refers to sugar. We have sucrose. Sucrose is your ordinary table sugar. Sucrose is going to be made of glucose and fructose. Fructose, a fruit sugar. And then here we have lactose. Lactose is going to be made of galactose and glucose. One way to remember that, lactose, you're going to find that in dairy products such as milk. Now, here's how I think about this. Okay, where do we live in the universe? We live in the Milky Way, what? Milky Way galaxy. So that's how I think of lactose is found in milk. We live in the Milky Way galaxy. So that's how I remember galactose, because it sounds galactic, right? And so, you know, we have that galactose plus glucose is going to give us lactose. Okay. And then maltose is going to be made of glucose and glucose. Okay. Now, the thing about disaccharides is these are sugars that we can get energy from rather quickly, but they have to be broken down in order for us to use them. Now, you might be or you might know of someone who is lactose intolerant. Now, how do we break these things down? Is we use an enzyme. Okay, and in this case, if you don't have enough lactase, and anytime you hear the word taste or the ending taste means one of two things. One, you're either at a hardware store, or two, it's an enzyme. Well, guess what? First of all, this ACE is spelled wrong for an enzyme, but we're talking biology, so it is going to be that enzyme. So lactase is going to be the enzyme that is going to break down lactose. And many humans either lack lactose, or as you get older, you start to produce less. And you're going to find out that if you're not breaking this down, it's going to stay in the gut, and it's going to act as a solute. And solutes, my definition of a solute is a water magnet, and that's going to hang on to water. It's not going to be allowed to absorb in the intestines. And if we keep water in the intestines, what are we going to wind up with? Diarrhea. Also, the fact that we still have a sugar in the gut, bacteria are going to feast on it, and we're going to wind up with gas as well. So, not a great thing. Okay, moving on. These are polysaccharides, okay, which means many sugars. And couple of the important ones here, we have starch, and we also have amylose. Amylose is one type of starch, amylopectin, another type of starch, okay? And the thing about starches is they can be broken down rather quickly to be used as energy, to get that glucose out of there for the cells to utilize for energy to produce ATP. And the enzyme, since we were talking enzymes, to break down starch is amylase. And amylase, one of the places it's found is in the saliva. And so we actually start chemically breaking down food, starch in this case, in the mouth. So chemical and mechanical digestion are taking place almost immediately. And we'll get more into the difference between chemical and mechanical digestion when we get to the digestive system. But again, amylase is going to start breaking down that amylose and that amylopectin. The other thing is glycogen. Now, as you eat carbohydrates, the excess glucose is going to be turned into, one of the things that gets turned into is glycogen. Now, what doesn't get turned into glycogen can then be kind of rearranged and turned into fat. But let's talk about glycogen. Glycogen is found in the muscles, and so that's one area that glycogen is stored at. And the other area is in the liver. And again, this is something that can be broken down right away by the enzyme glycogen phosphorylase. Again, we hear that ACE, A-S-E, not A-C-E like the hardware, but A-S-E. And so, as your muscles are working and they need more energy, for the ATP, then the glycogen, muscle glycogen, gets broken down right away to provide that extra energy. In the liver, that helps to regulate your blood glucose levels. So you ingest a meal, your blood sugar goes up, insulin is released by your pancreas, that lowers blood sugar. Okay, but throughout the day, that blood sugar goes lower and lower, and one way to raise that blood sugar up again, because we're trying to maintain a a steady blood sugar level throughout the day. Think about a teeter-totter or seesaw. Okay, if one end is up, that means your blood glucose levels are up. As you're utilizing the blood glucose, picture that seesaw tipping down the opposite direction. The idea is we want to make it level. So there are hormones that are released, and one of those is glucagon from the pancreas that tells the liver to release or start breaking down the glycogen back into glucose. And again, that's going to help raise the blood sugar. Okay. So again, we'll talk more about that in detail. I'm just kind of glossing over it right now, but when we get to the digestive system, we'll get into a little more detail about it. And then we have cellulose. Now, to give you a good example of cellulose, take a look at something made of wood. That wood is made up of primarily cellulose. Paper is cellulose. That part of a plant that you can't digest, that is cellulose. The cell walls of plants are made of cellulose. Okay, so what good is cellulose for us then if we can't digest it? Well, that is what acts as a source of fiber. Okay, and fiber is pretty important in the digestive system for doing a few things. One, it helps, of course, with the formation of fecal matter in the intestines, because it'll hold on to moisture and what have you. But the other thing is the normal flora in your gut also relies on cellulose as a fuel. So your healthy bacteria is going to, in your gut, again, is going to rely on a lot of that cellulose for it to be healthy. So eating fiber is good for you. And of course, nothing in excess, but we tend not to eat enough of it as is. Now, here's an example of triglycerides. Now, triglycerides are composed of glycerol, and we can see glycerol right here, attached to 3 fatty acids. Here's the 3 fatty acids via dehydration synthesis. Remember dehydration synthesis, where we take a hydroxyl and we combine it with a hydrogen, okay, and that gives us H2O, water. Okay, and so if you notice that the glycerol gives up the hydrogen atoms, so we have HHH, okay, and then each fatty acid is going to give up a ho. I'm sorry, not a ho. It's an OH. That's a hydroxyl group. So each fatty acid then is going to give up that hydroxyl group. And so these combine, and again, H2O, which is going to be water. Okay, and since there's 3 H's, 3 hydroxyls, when they combine, we're going to get 3 waters as part of the reaction. And so this is one triglyceride or neutral fat. Now, we hear all the time about saturated fats and unsaturated fats, and what's the difference? Well, saturated fats have a straight chain, and saturated fats tend to be solid at room temperature, whereas unsaturated fats have a little bend or kink in it. Okay, and if they're bent or kink like this, then they're not going to pack together very tightly. Here, let's go back to saturated fats. Think about those as pencils. If we pack pencils into a box, that's pretty solid, right? And now jam your hand down into those pencils and try to touch the bottom of the box. Okay, your most likely going to break a fingernail. If you did it hard enough, you might break a finger. Okay, so those are strong. Okay, so unsaturated fats, because they're kind of bent. Think about those packing peanuts, which usually wind up all over my house. But they have that little bend in it. And so push your hand down into that and it just kind of moves away. Well, those packing peanuts are not liquid. Molecularly, though, because of that bend, it's not going to allow the fats to pack densely. Okay, it's going to make them looser, hence a liquid. So other important lipids, we have phospholipids, and they're composed of two fatty acids, glycerol, and a phosphate group. And those are going to be important because this is what is going to make up our cell membranes. Okay, so that's why this is important. We have this phosphate or polar head. Okay, and if you see here, not the best pictures, but right here is that polar head. We have a glycerol backbone, and then we have these fatty acid chains. Okay, so remember those because we're going to talk about it in a little bit more detail when we get to the cell. Okay, and then we have sterols, and they're ring-shaped lipids. And what we see here is cholesterol. And then we have, that's right here. And then we have prostaglandins down here. Prostaglandins are derived from unsaturated fatty acids. And prostaglandins are important as inflammatory chemicals in many cases, such as prostaglandins E2 or PGE2s. And these PGE2s are going to be quite inflammatory. And this is actually what aspirin blocks the formation of. Okay, because again, these are pain chemicals, inflammatory chemicals. And if we block the formation of it, then we have an anti-inflammatory. And again, that's what aspirin would block, is the production of these PGE2s. Now here's the structure of an amino acid. Amino acids, when they're put together as a chain, we make proteins. And amino acids have an amino group. They have a side chain, and where you see R, this is variable, and this is what's going to make the specific amino acid that we're looking for. And then we have a carboxyl group right here. And how we make chains of these amino acids is we create what are called peptide bonds. And so when we make a long, what we call polypeptide chain, that then is what's going to be folded up and become a protein. So peptide bond, different amino acids join together to form peptides, polypeptides, or proteins via dehydration synthesis. The bonds between the amino acids are peptide bonds. So here we go. And now looking at shapes of proteins. We have the primary structure up here, which is our polypeptide chain. But now we're going to do a little origami on it and then fold it. And this is what's going to give the structure and function of specific proteins, is how it's folded. And here we can create an alpha helix or a beta pleated sheet, and this is going to give us our secondary structure. Now if we take it and we fold it again upon itself, we can wind up with a tertiary structure. And if we combined tertiary structures, two or more tertiary structures, we get a quaternary structure. And an example here would be hemoglobin, which is what is going to carry oxygen in your blood. Now, we've talked about enzymes a little bit, and remember they help to lower activation energy. They help to do things that the cell just normally wouldn't have the energy to do on its own. And it's either going to put things together or take things apart. And so steps in an enzymatic reaction, the substrates approach the active sites on an enzyme. So here's the enzyme. And it's kind of one of those lock and key type deals. If these don't fit exactly into the active sites, it's not going to work. So that's why we have specific enzymes for specific jobs, basically. In this case, we have two substrates, and they are going to come together and bind to the active site, and that is what's going to produce an enzyme substrate complex. Okay, so we have an enzyme substrate complex. And then the changes internal to the enzyme substrate complex facilitate interaction of the substrates. And as you see, this fused these two substrates together to form a product. And the products are released and the enzyme returns to its original form, ready to facilitate another enzymatic reaction. Okay, but we could also do this backwards. Okay, this could be our substrate. Okay, it can attach, okay, and then be split apart, and these would be our products. Okay, it just depends on what enzyme you're talking about and what the end result is that's needed. Now we're going to take a look at nucleotides. The building blocks of all nucleotides are one or more phosphate groups, a pentose sugar, and a nitrogen-containing base. Now the nitrogen-containing bases of all nucleotides are going to be these, which are cytosine, thiamine, uracil, adenine, and guanine. Now cytosine, thiamine, adenine, and guanine are found in DNA. We don't find uracil in DNA. Now in RNA, uracil is going to replace thiamine. So we're not going to find thiamine in RNA. We'll still find cytosine, adenine, and guanine, as well as uracil in RNA. Okay? And our two pentose sugars are going to be deoxyribose, again for DNA, ribose for RNA. Okay, DNA. In the DNA double helix, two strands attach via hydrogen bonds between the bases of component nucleotides. So you can see here our red is adenine, our yellow is thiamine. Now really that's all you have to remember. A combines with T. Okay, because the next two are guanine and cytosine. Okay, guanine and cytosine. So G and C combine. Okay, so G and C here combine. And A and T combine. So again, if you just remember A and T combine, that if I say G, well, it can't be T that combines with it because that combines with A. So G and C will always combine. Now, when we're talking about RNA, again, that's a different story. RNA is not going to be a double helix. It's going to be a single strand. And again, instead of finding thiamine on that single strand, we're going to find uracil. Okay. And again, we'll talk a little bit more detail later about how DNA actually works and what it's for. Now, the energy currency within a cell is going to be this ATP, which is adenosine triphosphate. Okay, so adenosine is going to be made up of an adenine group and a ribose. Okay, so adenine and ribose are going to then combine with 3 phosphates, and this is going to give us adenosine triphosphate. Okay. Now, how does the cell use this as energy? Well, these bonds in between the phosphate are high energy bonds. And if you break a bond, then let's see what happens. We're going to wind up with the release of energy. Okay. And so now we end up with what's called ADP, or adenosine diphosphate. So remember, it was triphosphate because there were 3 phosphates here, but we just released one of the phosphates and broke one of the phosphates, I should say, and released the energy from that bond. Okay, leaving us with ADP. Here's how I think about it. This is like a rechargeable battery. ATP was the charged battery. We needed energy from it. That energy was released. Now the battery needs to be recharged again for us to use it. And so how do you charge up a battery? Well, usually you plug it in, right, to an energy source. That energy is put back into the battery to recharge it up. Well, we're going to put energy in, and again, we'll talk about this more in detail later, but we're going to put energy into this ADP to phosphorylate the ADP back into ATP. So what does that mean, phosphorylate? We're going to take a phosphate and add it to ADP. Oh, by the way, this phosphate that's released can phosphorylate other things as well to form usually a little more unstable bond. And but again, when that phosphate breaks off, that gives off a little bit of energy, again, required to run some cellular processes. Okay. But again, if we put this, put some energy into it, we can reattach that phosphate. And now we're back to ATP again. And that's a process that takes place within the mitochondria of the cell.